Yeah, we can hold bar going again. That's just one of those.
Ela vai conseguir quando ela tiver.
How this little bar is hold bar again. If I keep the mouse over there, it'll go away on its own.
Do you think?
Então,
Yeah, so-
tem que fazer 50 primeiros de dezembro, né? Então, a gente encontra.
I think I actually.
Começa em
Um
setembro.
ano e meio, porque um ano e meio. Não, é 2 anos, são 2 anos. Mas é verdade, quando me pensa. Mas eu consigo trabalhar um pouco, assim.
That's Saturday.
Igual a escola, tipo, daqui não é de vai fazer.
Monster Mash.
Pode fazer uma reunião aqui.
There's definitely gonna be some just there.
There needs to be Monster Mash. Does just do the Monster?
Uma coisa assim, bom.
Não, eu pensar que pode ser de pegar tem drag assim, possibility de live.
I just found out it was like lots of people in the house, you know?
Get scared, but like, but not too bad.
Most cat, I mean, mine do kind of too. Matilda's a little bit brave, but like, roughly.
She'll come out, if there's like a quiet period.
Especially when.
Ai, gente, estou ansiosa para os perfis.
That was so funny and so sad. We did the rodent of unusual size run down the upstairs hallway of Roswell. Coming up this.
Nossa, acho que foi uma
I've never seen that guy move so fast in his life. He was,
off de
he
carne.
didn't come out from under the couch for like half an hour.
Maldito, ela tem em cima da minha carne que eu tenho. Toda chique, que eu comprei pra esfirra. A prévia.
Ah, não,
Ai,
faz quibe, muito melhor.
Não, tá bom. Vamos fazer, então.
Nossa, que sai. Fonte.
Como isso acabou? Esses minutos são os piores, né?
Péssimo.
Eu odeio essa espera. Teve uma época que eu comecei a chegar atrasada com as coisas, gente. Eu falava, só pra não ter esse minuto de espera, assim, sabe? Pra chegar, eu tinha que ir.
Sei. Ó, eu acho pior, né? É congresso.
Isso, do nada, que vem aí.
Tem aqui, tipo,
Around the room anyway.
So like my son was dodgeball.
Mhm. Fuck you. But listen, like,
E eu ainda tenho exercício para fazer, né? Eu travo a língua, eu tenho que destravar, então já mas é, eu faço, eu aprendi na comunidade aula, na escola, uma vez foi uma fome super legal, na trabalho da gente. Gente, muito importante esses negócios, mas a voz uma hora pra ele. A gente tinha que ser a voz, a gente ia começar o dia, porque pensa, você acorda, aí eu dirigia assim, entrava, até chegar na escola, no primeiro carro, chegava lá, 10 para 7, entrava às 7, ó, até 1h30, sem parar. Aí eu, isso todo dia, até o terceiro. Não é só começar o ano, é terceiro, isso não vai ser.
Now that I got my chair. Hey, hey. How was your day? You had fun?
Vendo agora.
Oh, yeah. I was like, what? How was your fun? It was a lot. I had.
será que aquela da planta não funciona?
O vídeo não. Ela tem outras coisas para planta. Por exemplo, a nossa planta que não tem
Ah, It's, she's having a little. Because I feel like celebrating the weekend. Before I got but that's the great weekend after.
I was imagining just.
Eu não tenho
Yeah.
nada, gigante. Eu também não tô olhando. Então, só que essa daí é muito pequena, sabe? Tipo, quanto maior, mais tangente você tiver, é melhor, porque você tem uma coisa que tem um pouco de luz aqui. A tua periferia ainda vai estar escura. Você pega um negócio desse tamanho, planta a cara, você vai te deixar lá no meu comprar uma outra pra deixar em casa. Mas não sei, não tá. Força que essa pequena você tem que andar com ela assim, ó. Quanto menor, mais perto de você ficar, né? Mas acho que dá pra começar com ela. Vou olhar, já vi isso aí, essa coisa.
Does that start?
Muita coisa até. Vou tentar pegar usada só porque já não vai.
Whoa, Nancy.
Queria gastar 100 dólares com vou precisar de duas.
Spirit. I was going.
Não, eu vou deixar em casa. Eu vou deixar aqui.
Tragic.
Eu vou deixar aqui. A música mais com aquela 15 a 20 minutos por dia.
Nothing more. They're gonna get that's totally changed one of my minds. Yeah. A regular.
Just everyone should have something to do. In March, I did a meeting, and so it was sad that she.
No, they like send me. Since we were sitting here, I was like, let's try about 16 o'clock.
It's, I mean, now.
Yeah.
Yeah.
Oh.
That's awesome.
It is.
Drum roll.
Incredible, yeah.
That's her rating.
For the money work. Is that really what?
Ela nunca me contou isso, né?
Exactly.
Great.
Same.
It's just-
Yeah.
I'm going to ask for us.
Oh, yeah.
Is that what you're gonna
What's that?
Yeah, it's kind of. We're gonna do it all at my house, so.
I know there's a difference.
It goes towards like buildings.
Okay.
Tax and insurance, but also.
Oh, yeah, I was hard to.
And I can do this slow other than one or like major. Great, I'm really going.
Good afternoon, everybody. I think we're gonna go ahead and get started now. So for this week, we have Marianne Silva here, a research scholar with EEB, to talk to us a little bit about biodiversity and neotropical Savanna.
Thank you everyone for being here today, and also thank you everyone that's watching over Zoom. I'm Marianne, I'm a postdoc in Marjorie Weber Lab, working with mites and plants in mutualisms, trying to understand the evolution of this interaction. But today I'll not talk about my postdoc. I will talk about the work that I developed during my PhD in Brazil. So today, next minutes, we'll talk about another group of small organisms. We'll talk about what I'm calling the small giants. So when you look to this title here, three words came up to our eyes: and biodiversity and neotropical savanna. So based on this, by the end of my talk today, we will try to answer these three different questions. So why study ants? What's so special about the neotropical savannas when you are talking about ants? And what did I discover during my PhD with the ants from the neotropical savannas? So let's start from the beginning. There are different ways that I could start, but I'd like to start just to how beautiful ants are. So how cute an ant can be when preying some termites. Or how beautiful they are cutting some leaves. Wow. By the way, if you don't know the website of Alex White, take a look. The colors, how they are different in their colors, like this golden, goldish color here, or the textures in their bodies, or even the different shapes they have. How is the polymorphism within the same species? So here you can see a soldier and a worker from the same species. Or how they organize themselves inside the colonies, dividing their work there, how they interact with intra-specifically and also inter-specifically, and also how they interact with other organisms such as plants, trophobionts, or even how they can be used as reference for some other organisms. So here you can see a spider that mimics this ant here and also prey this ant here. I wish this could be enough to advocate in favor of ants and say how they are great and beautiful and perfect is the system. But I know that's not enough. So let's take a look at some data about ants. If you look to this map here, the colors represent the number of ant species. So hotter the color, the higher the number of ant species, we will see that ants are almost everywhere. So I bet that if you have ever been in a field course, probably someone made a project with ants because they're easy to find and easy to work with. But more than being widely distributed, ants also comprise a huge biomass. So they represent a huge amount of the biomass of the heteroarthropods. Just to give you a reference, this is more than the biomass of all mammals and birds on Earth. And if you compare to humans, they are about 20% of human biomass on Earth. So they are a lot. Also, the last estimate about the number of ants on Earth, it's about 20 quadrillion individual ants on Earth. So, given they are widely distributed, a lot of biomass, high number, we can expect them to have a huge impact on their ecosystems. So, controlling for entomological control, soil turnover, plant germination, seed dispersal, or even pollination. And if you take a close look to this graph here, you see that the great amount of the ants that live above the soil, the FJ ants, are found on the tropical areas. Specifically, most of them are found on tropical savannas. So here is our connection to the neotropical savannas. And we can go to our second question. Why study ants in this biome? So Cerrado, that is the neotropical savanna, is considered one of the world's hotspots. It covers around 26% of Brazilian territory, but only 8% is under legal protection. So it's a biome highly threatened. One of the explanations about why Cerrado comprises a huge ant biodiversity is about environmental heterogeneity. So in Cerrado, we can find a range of vegetation types going from open grasslands, passing through savanna-like formations, and up to forest-like formations. And this is in a response to fire incidents and also an inverse gradient of soil fertility. So it's believed that this huge biodiversity boosts ant diversity. But also on Cerrado vegetation, ants can find a lot of resources. They can construct their nests inside the plants, and also about 30% of plant species present extrafloral nectaries in Cerrado. And about 30% of plants present interactions with trough biomes, these small insects here. So this means that on Cerrado vegetation, they can find a lot of sugar. And once they are on vegetation, they start to protect their resources, and they increase the defense against herbivores and also the protection of trophobiomes against predators. So if you are interested in ants, you may want to go to Cerrado and take a look on ants there. So what I discovered about the ant biodiversity? First, let's define biodiversity. According to the last definition, biodiversity is the variability among living organisms from all sources. These include variation in genetic, phenotypic, phylogenetic, and functional attributes. In other words, we can investigate biodiversity using different tools, genetic, taxonomic, phylogenetic, functional, and look to different biological levels of biological organization. About the distribution of biodiversity in the planet, Abby showed this in the last seminar. The most consistent pattern of distribution is the latitudinal gradient. So here in this image, you can see that hotter colors represent higher diversity, and its diversity is more concentrated closer to equator. But for some groups, this pattern can be different. And this is the case of the ants in the Cerrado. So here you can see that in higher latitudes, away from the equator is where we find more ant diversity. So I asked, can we observe this pattern for other levels of biodiversity? And this was the main objective of my PhD, to investigate the ant biodiversity along the latitudinal gradient of Cerrado. So I look at to the diversity about the communities, interested about the interactions between ants and plants. And I also look into the population level, thinking about the functional variation and also genetic variation. But more than describe the pattern, the spatial distribution patterns of these levels of biodiversity, I also, this became the 3 chapters of my dissertation, but I also wanted to try to unveil potential abiotic and biotic factors underlying these patterns of biodiversity. So here is our sampling scheme. In the beginning, we had another point here, but we lost this for the fire. So We selected seven sites in the Brazilian Cerrado savanna, all of them with the same vegetation type. In each site, we established 5 transects of 200 meters. We have to divide these transects in 20 parcels of 10 meters, and was because we realized it would be a lot of plants to sample, so we have to adjust the sampling design. And in each parcel here, we collected all information about all plants. So in total, we analyzed more than 3,000 plants, and which kind of data we collected. So each plant was observed, was measured by height, diameter. We characterized the vegetation density. We saw presence of extrafloral nectaries or trophobiomes. Each plant was observed for ant interactions. So we counted the number of ants, the presence of ant nests. We identified the plant and ant at species level whenever it was possible, and also collected abiotic data. We analyzed the soil and we obtained other climatic data from public databases. So with this, I'll show you the first chapter of my PhD about the interactions between plants and ants and plants. So we know different variables can influence ant-plant interactions, such as latitude, plant species, environmental heterogeneity, abiotic data such as water, the plant species richness, and also the plant life history. But more than that, we know that these variables here influence each other in different ways. So we ask, is there some pattern of geographic distribution in the frequency of ant-plant interactions in Cerrado? Our hypothesis is that yes, following this, we expected to find a higher number of plants interacting with plants as higher the latitude. And about how different biotic and abiotic factors influence this interaction, we used structural equation modeling. So for this kind of model, you have to hypothesize how different variables are related to each other based on literature. So here is our initial hypothesis. So latitude would influence seasonality, that in turn influence soil, organic matter, plant community, and fire frequency. The fire will influence plant community, vegetation structure, and ant community. Soil will influence vegetation structure and plant community. The plant community will be determinant for the resource availability on vegetation. And then the resources and ants will influence the amount of these interactions on vegetation. Good, here is what we found. I have to change this graphic, but just remember here we are in the south. So here it's far away from the equator and here close to the equator. That's why the negative values of latitude. So we indeed found a negative relationship. So far away from the equator is where we can find more plants interacting with ants. But okay, so how our structure equation modeling can explain this? So here was what we found. So the red arrows are the negative relationship, and the black ones, the positive relationships. And we can see that indeed latitude will influence the interactions through the impact on seasonality. And also one thing that was interesting is that the amount of organic matter in the soil negatively influenced the amount of resources on vegetation. And unexpectedly, the resources influenced ant community, but was the ant community that is directly related to the proportion of plants interacting with ants. And if you take a look close to the resources, this you have to believe me, this is just a schematic, not numbers. But what really matters for this interaction is the sugar-rich resources, not the nests. But if you want to see some numbers, let's do it. So based on this, you can estimate the direct, indirect, and total effects of each of these variables on ant-plant interactions. And you can see that seasonality, then resources and ant community have the highest effects on ant-plant interactions. But looking carefully to the variables that directly influence these interactions, you see seasonality and ant community. And between them, ant community has the highest effect. And this is the main lesson of this first chapter. So if you are interested in work with ant-plant interactions, you should include the ant community in the studies. Maybe it's more important than just quantify the resources on vegetation. So you can go to the second chapter, where I look into the functional variation, but not in a traditional way between species, but within the same species. So we know that how individuals thermoregulate is gaining research attention because of global warming. And ants can be a good system to study how species will respond to global change because they are almost like plants. They cannot move so much. So because they are attached to their nests. So they have to acclimate. They have to respond to environment. And indeed, ants have a huge phenotypical plasticity. Moreover, they are widely distributed, so we can investigate ants under different environmental settings, and this can give us a clue how the ants will respond to global change. There are different ways to do that, but we can use functional traits. In ants, there are two traits related to thermoregulation. The pilosity of the mesosoma, that here is the number of hairs that cross the mesosoma profile, and also the body size. So both of these traits are correlated to different thermoregulation characteristics. And you also know that different factors influence this variation. So if you could express multiple potential factors that influence the functional variation, this could be interesting. So to study this in the Cerrado, we choose this ant here. This ant is one of the most abundant ants. This species is almost in all, I think all of our transects, this ant is present. And the name is Camponopus crassus, and the sugar-rich resource count for about 8% of the diet of this ant. But more than that, the real reason to choose this ant is because we observed that they have huge phenotypic variation. So here you can see an ant with a lot of hairs in the mesosoma, and here almost no hairs. So we asked why this variation happens. And we tested this in response to different predictors, including community, environmental characteristics, plant community, and resources. And for body size, we didn't see an effect of these predictors here, but for pilosity, we found the resources is the most important variable associated to the variation in the pilosity. And interesting, you can see that it's a negative effect. So which means that the higher the sugar-rich resources availability, lower the velocity. And if you look close to the effects of the resources, we will see that the proportion of plants with tropical biomes and extrafloral nectaries are the ones that most account for this response here. And maybe this is my favorite part of my PhD, because there are two possible explanations for this. The first one is about behavior. So we know that Camponotus crassus during the rainy season, the foraging area, it means the distance they go far away from the nest ranges from 1 to 3 meters. But during the dry season, when the resources are more scarce, they can go further. So it means that they can adjust their foraging depend on the amount of the resources. So probably in that places with lower resource availability, they have to spend more time outside the nest. So they're more exposed to external conditions, and maybe increasing the pilosity. It's a way to thermoregulate, to increase thermoregulation. But the other explanation that I prefer is about a metabolic explanation that I discovered just after I obtained the results. So it was. So sucrose is used to produce heat shock proteins. So it's known that for some insects, sugar-rich diets increase their activity, their thermal limits, and their thermal tolerance. So given the sugar-rich resources accounts to up to 8% of Camponotus crassus diet, maybe it's the main resource for thermoregulation and for increasing their foraging. So can be, we hypothesize it, that in places with lower resources, maybe this species has to invest in a different strategy of thermoregulation, maybe increasing pilosity. So one of our hypotheses is that some kind of trade-off can be happening here. So here was the second main lesson of my PhD, that resources may mediate species response to global change. So if you are interested in understand how species will respond, more than see the response like for the species itself, maybe also better to consider the resources they are using and how change will affect the resources. So with this, we can go to the third chapter of my PhD. We know that this pattern that I showed you exists for species diversity, and almost all the works are about species diversity. And we still lack information about the distribution of genetic diversity. But it's believed that this pattern is similar between these two levels of biodiversity. And indeed, some workers have shown that these levels of biodiversity are correlated because they can respond to similar evolutionary forces, like migration can bring new alleles, but also can bring new species. Drift can lead to random fluctuations on alleles or on species. And selection you can select alleles or species. So this can be a parallel effect. On the other hand, you can also observe casual effects. So populations with higher genetic diversity are also more prone to speciate, and also with communities with more species, mainly in the context of competition. In these communities, new phenotypes and genotypes are more likely to arise. Moreover, we know that environmental heterogeneity is linked to genetic diversity, can be through natural selection or mediating demographic processes. So, knowing this and knowing that this pattern is found in Cerrado, and I showed you that we also have this pattern for ant-plant interactions, and other works show that they also find this pattern for network diversity and for vertical stratification of ants, which means their position in the plants. So I asked, how about the genetic diversity? So this pattern can also be found in the most fundamental level of biodiversity. And the answer is yes. So we also use the Campanotus crassus to investigate this. You can see here that we use SNPs, and for 3 different estimates of biodiversity, we can find a clear pattern of an inverse gradient. When we look to the potential effects of biotic and abiotic factors, we found that precipitation is one of the factors that explain this pattern. But what's about precipitation? Precipitation is highly associated to ant nuptial flight. So we know that for some species, nuptial flights are less likely to occur during dry season. But we also know that for other species, nuptial flights can be favored under hot and dry conditions. So we know nothing about the reproduction of Campanotus crassus, but this can, if they are, let's suppose, if their nuptial flights are favored under hot and dry conditions, and this increases their genetic variability, this can be important if you think about the predictions for the Cerrado, that the predictions say that Cerrado will become more hotter and drier. So maybe this can be good for the maintenance of the genetic diversity in this species. And also, when you see here, another important factor that influences the genetic diversity of Campanotus crassus is the amount of sugar-rich resources. So higher the availability, higher the genetic variation in this species. So probably because we know that resources may mediate the demographic process. We know, for instance, that ant colonies grow faster under high resource availability, so these can be mediating demographic processes. So the third lesson was that distinct levels of biodiversity present the same latitudinal pattern. And the interesting thing is that we have 7 sites, and we recover this pattern in 7 sites. So it's really strong. What's behind this? We still don't know. We had clues, but the mechanisms we don't. Great. So I think the main lesson of everything, mainly thinking about our work completely based on field collection and observation, is that everything can start with a really simple question and observation. And in the last years, I started to advocate in favor of natural history. So I also bring some things to think, mainly now in this new era of science, of fancy models. I think we need to back to basics and look carefully to natural history again. So if I could say something, I would say that simple and opportunistic observations can open new research avenues. if you are a natural history enthusiast, please publish your observations or try to archive them in public repositories. So for instance, for ants, we have EntoWeb that you can add your observations. So please do that. If you are a PI or a mentor, natural history is a great avenue to introduce younger scientists to science. So it's easy to do, it's not so expensive, it's a way you can make a research driven by questions using natural history. Also, always support researchers with natural history. It means like, if you work with models, talk to people that work with your system in the basics. Talk to them. Maybe the answer is not about weather or fancy things. It's about where they live, what they eat, with which other organisms they interact with. And finally, natural history needs all you can give. So make the effort to include natural history data on your projects, manuscripts, and talks. So let's suppose you're gonna talk about, I don't know, you saw a lot of talks about fish. Please give us a background about where they live, what they eat, which kind of interactions they play. It's important. And also in your manuscripts, if possible, I know that sometimes we have to reduce the manuscript, but include a part about the natural history of your system. And these can be an action to overcome the huge decline that we are experiencing in natural history. And this is particularly important for us that are working in the tropics, because we still don't know a lot of our natural history, and sometimes it's hard to publish this data. So all the effort is really welcome. Well, with this, I'd like to thank you, all my collaborators, and mainly this professor here, the Paulo de Oliveira. He's one of the major ant experts in the Neotropics, and I had the privilege to be his student since undergrad. I did also my master and PhD with him. I also have to say a huge thank to my previous university in Brazil and the financial support that I received. And with this, I would be happy to answer any questions.
That was really cool. I was interested in the spatial decline you saw in genetic diversity. And I was curious from the chapter 1 data, can you estimate the density or the abundance of Uskempenor's crassus?
Yes.
From those data and see how they're correlated with diversity? Like, is it just abundance and diversity or like a really tight correlation, or is it messier, or can you not get
I
those
did
data?
this.
Oh, you did?
Yeah.
What did it look like?
No relationship, no significant relationship. Yes. I need to talk to you about this.
Well, yeah, that's cool.
Yeah, I tested different things, and one of the things was the abundance of these ants and also their frequency on vegetation. So it's similar but different. The abundance, like literally the number of the ants, and the frequency is the number of plants with these ants. Because given ants are social, so sometimes we have to consider just their presence and not their number. So I tested both and no correlation, significant correlation.
Great talk. It's really fun to see your research.
on the big screen. I was wondering whether, and maybe you mentioned this, but do plants that produce like extrafloral nectaries or like the trophobionts, like did the proportion of plants that have these things vary in the Brazilian Cerrado, like in a way that might explain like why they have this like reverse latitudinal?
I'm also working in this, undergrad in Brazil, did like a project with this, and no. We don't have a latitudinal pattern for the fence. So we are now working, because it was a lot of data, so with Marjorie, I now want to investigate the phylogenetic part, but we didn't find a phylogenetic pattern for the plants, but I still don't know about the ants. I need more time to work on this.
How has the Cerrado changed over the last millions of years, and could that explain some of the patterns you've seen in terms of the age of the habitat?
Yeah, I think this data collection was from 2013 and 14, if I'm not wrong. We lost almost all areas. So I'm talking about the past here. So most of these areas here burned, but not natural fire, criminal fire. So we even like market all the plants to back in the future. We don't have this anymore because we lost everything. So I believe this can be, I know that the works that I showed you today, it's like a photography, like it's not a video, you know. So it's about a given time, and I do think that this change in Cerrado would influence this. I really would like to back and see like for genetic variation, if there is something different. But I also know that given Cerrado, we have these natural fires. Cerrado has this ability to recover. So maybe this pattern is strong enough, or it's like resilient enough, but I don't know.
Thinking about like what's the future of that habitat? Is it obviously it's getting burned down, but is it expanding or is it contracting? Like you're changing?
Yeah, I think there are two different ways that Cerrado is going. The first one is reducing areas because of like agriculture, and the second thing is that people are afraid of burning in Cerrado. So some conservation areas are protect Cerrado against burning. And this is increasing the biomass in the Cerrado. So some areas that are open grasslands or savanna-like formations are becoming forest-like formations. And although we have this increment in biomass, we are losing diversity. So we know that this increment in biomass is leading to a decrease. For example, one of the groups that the researchers studied was ants. So we are losing ant diversity. It is in this increment of biodiversity in biomass. So also we have this problem because people don't want Cerrado to burn, to lose areas, but Cerrado needs to burn to maintain its diversity. Is something that, and we don't know nothing, for example, about the genetic diversity in this context. Yeah.
Great job. I was wondering, you said that, as you get more biomass, you get the more forest-like structures, that it decreases diversity. And Julia asked about the, presence of ants or of plants with external nectaries and trophobionts. And you said that didn't have an effect on ant diversity. But does the sort of like vegetative structure, like, do you see that there's a latitudinal trend in that, where there's more forest-like things, maybe towards the equator as you're seeing diversity decrease?
Yeah, this is a good question because we focus on all our sampling sites are the same Cerrado vegetation, the savanna-like formation. So we don't know about if we could find different thing and a different, like in a forest-like information. Because previous works with latitudinal gradients also focused on savanna-like formations.
Okay.
So, yeah.
So we don't know about the
We don't.
vegetative variation.
Is there any data in the Atlantic forest? I know it's like a different place, but is it more forest formation?
Yes, we have data. We have a huge data set about the ant diversity in Atlantic forest. And we also have an inverse latitudinal gradient.
That might
For the
answer your
Atlantic
question,
forest.
because it's totally forest.
So it's something really strong.
That natural history question.
Please.
You showed that really striking figure that showed what, like 30% of plant species hosting triple biomes and 30% hosting or creating extrafloral nectaries. And do you find that ant species tend to use both? Like what is the overlap in resource use there? Do you get species specializing on one or the other?
This is super cool. Most of ants prefer the resources from tropical biomes. Yes, we have different works with that, and almost all of them show that ants prefer the exudates of. It seems that the chemical is different, and if you look to the results of my functional data, the predictor that most contribute for the tendency was the proportion of plants with tropical biomes. So, and they have like clues of each plant has tropical biomes. So it seems that they drop some liquid in the soil and ants realize that and go to the plant. Following them.
Did you learn that from like choice assays with ants and different nectars?
No. I know, like one of my collaborators made this, field observations showing that, but like, no, I don't know if there is some work with it. Good, very cool.
I thought the hands up.
I don't know. I don't know.
You may have already talked about this, but how many species can to co-occur? And is there a geographic structure in the number of species that co-occur and their interactions?
Like the ant species?
Yes.
Okay, yeah, they have some interspecific interactions, like really aggressive ants tend to manipulate the plant. So yes, it's something that I also have data to work on about the co-occurrence of ants. Yeah, I think it's a great idea. Like compare the data that we have from GB to the one that I have, like that are indeed interacting with the plants. This would be great work. Thank you. Thank you so much.
All this natural history data.